Bonjour à toutes et à tous et bienvenue à notre webinaire de clôture du projet Interreg ValBran. Donc vous êtes nombreux déjà à être connectés et nous vous remercions d'avance. Près de 150 inscriptions, donc c'est vraiment un succès. Nous sommes heureux de vous compter parmi nous. Dag iedereen en welkom op onze ValBran slot evenement met bijna 150 ingeschreven voor dit webinar. Het is zeer bemoedigend voor ons. Dank u. Donc, avant de passer la parole en fait aux orateurs, je voudrais simplement, en tant qu'organisateur de cet événement, vous rappeler quelques bonnes pratiques afin que ce webinaire se déroule pour le mieux. Je vous rappelle que ce webinaire se déroulera bien en anglais pour les présentations scientifiques, mais ici l'introduction, je la fais dans les deux langues afin que tant les francophones que les néerlandophones nous comprennent correctement. Voordat ik het woord geef aan de sprekers, wil ik u herinneren aan enkele goede praktijken, zodat het événement goed verloopt. Donc, bon à savoir, tout simplement, nous vous invitons à ne pas enregistrer ce webinaire, évidemment à ne pas diffuser cette présentation sans l'autorisation préalable des organisateurs. Je vous informe également que le replay et les présentations vous seront envoyés donc à la fin du webinaire, pas de panique. Neem dit webinar niet op. Gelieve deze presentatie niet te verspreiden zonder voorafgaande toestemming van de organisatoren. Replay en presentaties worden aan het einde van het webinar gestuurd. Bedankt. Quelques conseils d'utilisation concernant l'outil Livestorm. Vous connaissez peut-être certains, mais donc c'est un outil qui nous permet de tenir des visioconférences. Il y a plusieurs boutons d'action qui sont disponibles en haut à droite de votre écran. Vous pouvez le voir, il y a l'onglet chat sur lequel vous pouvez dialoguer, converser, mais pas poser vos questions aux orateurs. Pour ce faire, nous vous invitons donc à poser vos questions via l'onglet questions en haut à droite de votre écran, en précisant évidemment à qui s'adresse votre question. Ce sera beaucoup plus facile pour notre modérateur Jackie. Les réponses seront donc transférées également en compte-rendu des réponses en fin de webinaire avec le compte-rendu de l'événement. Toutes les questions, évidemment, ne pourront pas être abordées dans le temps imparti de la session des questions-réponses, mais voilà, on suppose bien que vous comprenez. Alors, pour le questionnaire de satisfaction qu'on enverra à la fin du webinaire, il sera accessible via l'onglet « Sondage ». De actieknoppen zijn rechtsboven in uw scherm. Om een vraag aan een spreker te stellen, kunt u de knop Question vragen gebruiken. U wilt de antwoorden op het einde van het webinar ontvangen. Er wil ook een tevredenheidsenquête zijn via het tabblad Sondage. U wordt uitgenodigd om de enquête in te vullen aan het einde van het webinar. Un petit mot concernant la langue. Donc, afin que tous les participants profitent du webinaire de la même manière et afin de permettre aux orateurs français de s'exprimer de manière confortable, l'ensemble des présentations scientifiques se dérouleront en anglais. Les présentations seront traduites en français et néerlandais, du projet ValBran évidemment, et vous seront envoyées quelques jours après ce webinaire. Dus voor de taal, om ervoor te zorgen dat alle deelnemers in gelijke mate kunnen profiteren, zullen alle volgende presentaties in het Engels zijn. De presentaties van de resultaten van het ValBran-project worden in het Frans en in het Nederlands vertaald. Ze worden enkele dagen na het webinar naar u toegestuurd. Et enfin, je vous promets, ce ne sera plus très long, quelques mots sur le programme Interreg France-Wallonie-Vlaanderen qui a permis en fait au projet ValBran de voir le jour. Donc il s'agit d'un programme de coopération territoriale européenne France-Wallonie-Vlaanderen qui a pour but de favoriser les échanges transfrontaliers. C'est un programme qui est soutenu par le Fonds européen de développement régional, le FEDER, pour des projets qui répondent à plusieurs thèmes de coopération. Ce projet-ci, donc le projet ValBran, fait partie de la catégorie recherche, innovation et transfert de technologie. Donc, comme vous le voyez à l'écran, il y a 4 thèmes de coopération. Het is een programma voor Europese territoriale samenwerking tussen Frankrijk, Wallonië en Vlaanderen om economische en sociale uitwisselingen te stimuleren tussen 5 grensregio's. Het is gesteund door het Europese Fonds voor Regionale Ontwikkeling. En het ValBran-project is een project dat valt in de categorie onderzoek en innovatie, technologie overdracht. Les opérateurs, comme vous le voyez à l'écran, alors ce sont des opérateurs qui sont français, wallons et flamands. Il y a l'Université de Reims, Champagne-Ardenne, l'Université de Picardie-Jules Verne. Il y a également le pôle de compétitivité français IAR. Jackie Van de Put nous fait l'honneur de sa présence aujourd'hui. L'ESBL Valbiome, Inagro, Vito, Jean Blou Agrobiotech, Université de Liège, Magali Deleu, ici présente, Catalysti et Greenwin. Au niveau des financeurs, comme je le disais, nous sommes soutenus par la région Grand Est, la province West-Vlaanderen, la Wallonie et le Fonds européen de développement régional. Je vais tout de suite donc passer la parole à Jackie Van de Put de l'IAR afin qu'il rentre dans le vif du sujet et afin qu'il vous présente les différents orateurs de l'après-midi. Dus ik geef het woord nu aan Jackie Van de Put. Dank u wel, merci pour votre attention et excellent webinaire à tous.
Thanks, Lauren, for this kind of introduction. So we are going to discuss today on the closing event of the ValBran Interreg project, which is a project which is dedicated to the synthesis and the assessment of surfactants for different applications, detergency, cosmetic, and plant protection, and also food additives, using some, I would say, biomass and for making local surfactants. And it really answers to the, I would say, COVID situation and the discussion we have today on the resettlement of biobased chemistry. So for this closing event, we will have the three, I would say, skilled lady, which has worked during 4 years on the development of the biobased surfactants. And let me introduce them. First of all, we have Professor Caroline Raymond, which is involved in the University of Reims Champagne-Ardenne, especially on the UMR-FAR, on the fractionation of biomass of crop. We have also Mrs. Dr. Magali Deleu, which is our expert in biophysics, which have worked on the correlation between, I will say, the structure and properties of the surfactants. And the speaker is also Winnie de Jong from Vito, which is a project manager, and which is working specifically on the process, but also on the technical and economical assessment of the project. And we will get also two, I will say, representatives from companies, Dr. Boris Estrin from the well-known biorefinery research center ARD, which is also involved in wheat oil development of surfactants, biobased surfactants, and Stéphane Verschave, which is a regulatory, I will say, director from a well-known also company, Sopura, in hygiene in general for food and other sectors. So you have received the program. So there will have two sessions. The first one will deal with the main outcomes from the projects. That means we'll cover all the value chains, starting from the hydrolysis of the biomass to the assessment of the different property and the also assessment of the economical aspect. We'll make then a pause, and after there will have a session of the different companies which are there, which speak more about the market, the existing biobased products and biobased surfactants on the market, and we'll discuss about also all the barriers to the markets. And I will take at the end of each session some questions, and we'll try to answer to all these questions. So let's have a quick word regarding surfactants. You know that because of their properties, their wetting properties, their forming properties, and solubilizing properties, surfactants are very used in a wide range of applications, not only in the cosmetic or detergency, but also in different industrial applications like plastic synthesis and formulation, like also ore mining or pen formulation. So many, I will say, application. It represents something like 1,200 kilotons per year worldwide. And if we look at specifically on biobased product, it's something like 2,400 kilotons per year, which are produced and used for these different applications, but specifically on for high value added market, niche market like cosmetic, especially in cosmetic, and also detergency, because it answers, I would say, to the customer needs, which needs performance, clean label, but also natural, I would say, advantages, but also because they have technical properties like better forming properties, like also the possibility to use it at lower temperature, higher pH, salinity, and with also a lower ecotoxicity, a good biodegradability. So that means many advantages for this biobased surfactants. And you know that since 2007, there is a rich the evaluation of chemistry substances, and there is some substances which are quite today very concerned, I would say, very toxic, and it represents a possibility to change them with biobased products. So you have here some figures, noting that the very high main, I will say, application detergents and the different type of applications in industry, on washing machine, on dishwasher. So this project deals with biobased surfactants, but you know certainly that there is also a existing range of biobased surfactants on the market, especially some sucroester and some alkyl polyglycoside. It represents something like 100,000 tons per year, which are produced. And there is also, I would say, a new development on ionic biobased surfactants, on microbial surfactants, or lipoamino acid surfactants. That means a lot of existing biobased, partially or totally made from, I would say, biomass. And 10 years ago, there's been a launch of alkyl polypentoxide from wheat oleo, and we'll discuss about this today. But the main innovation of the ValBran project is that not only we use enzyme to hydrolyze the biomass, but we use enzyme also to develop a new kind of, I would say, surfactants, and to functionalize the surfactants with also the development of a cascade of process, and in order to develop new properties to answer to the food additive, detergency, cosmetic, and plant protections, I will say, specifications. And also the advantage of the projects that they are covering all the value chains. That means if we look at the value chains, we're starting from the wheat bran, and we develop some surfactants and also make different applications. But also we are dealing on the cake and looking at the cake, I will say the cake brand is improved after it can be used to the other applications. So as explained, the first things that will be to make a focus on the main, I will say, outcomes of the project. And I will give the floor, first of all, to Professor Caroline Raymond, which is the coordinator of the project ValBran now. Thanks a lot, Caroline.
Thank you, Jackie. So, good afternoon. My talk will focus onto the development of an integrated processes to produce alkyl polyglycoside and sugar esters from wheat bran. This work was performed in 2 laboratories from Reims Champagne-Ardenne University, the FAR laboratory with the chair Affaires, and the Institute of Molecular Chemistry from Reims. The goal of the project ValBran is to valorize wheat bran into biosurfactants. As you know, wheat bran is an agricultural co-product from milling industries and from 3rd generation bioethanol industries. Its main use is for food and for feed. The strategy developed in the project is to catalyze enzymatic reactions to fractionate cellulose and hemicelluloses from wheat bran and to further functionalize the released sugars with fatty acids and alcohols. This allows the synthesis of alkyl polyglycosides and sugar esters, which are surfactants of interest for various applications. We also evaluate the use of wheat bran residues generated at the end of the enzymatic reactions for feed application. The project involves 9 partners, as described by Laurent during the introduction of this event. And on the scheme on the right part of this slide, you can see an overview of the organization of the project that is organized in 6 work packages. The first and the second work package concern the project management and the communication. The third work package concerns the enzymatic fractionation of wheat bran into sugars. The work package 4 concerns the enzymatic and chemoenzymatic production of biosurfactants. The purification and characterization of the surfactants concerns the work package 5. And finally, the work package 6 was focused onto the technology transfer with economic feasibility and environmental impact. Some few information about alkylglycosides and sugar esters, which are non-ionic surfactants. Alkyl polyglycosides, the world market of alkyl polyglycosides is more than 100,000 tons per year, as mentioned by Jackie. They are used as emulsifiers, foaming agents, wetting agents for cosmetics, detergency, and also phytosanitary. Commercial alkyl polyglycosides are produced by companies such as Wheatoleo and Cepic. The world market of sugar esters represents 10,000 tons per year, and they are mainly used as emulsifiers for cosmetics and for food. Croda and Cisterna are some examples of companies producing sugar esters. In our lab, we are developing biocatalytic processes to produce alkyl glycosides and sugar esters. The biocatalytic reactions occur under mild conditions, for instance, at temperature lower than 50 degrees. The biocatalytic reactions are performed in 1 step. Enzymes being highly selective, the reaction's products possess well-defined composition and structure. And finally, the use of enzymes as catalysts allow to limit the formation of undesirable co-products. So, producing biosurfactants with biological processes provides a global naturality of our final surfactants. Before starting the ValBran project, in FAR Lab and ECMR, we studied the ability of some xylanase to catalyze transglycosylation reactions in presence of xylans as donors and in presence of acceptor molecules possessing a hydroxyl group. In this example, the reaction occurred in water at 60 degrees. Various acceptors can be used, and when aliphatic alcohols are tested, the synthesis of alkyl polyxylosides occur by adding the aliphatic chain onto the anomeric position of the glycone part of the molecule. The glycone part of the alkyl xylosides can display a degree of polymerization from 1 to 7. An important feature of the transglycosylation reaction is that the reaction is radioselective and leads to the synthesis of anomer alpha or beta, depending on the enzymes. Another important feature of the reaction is that the transglycosylation is in competition with hydrolysis catalyzed by the xylanase, as the reaction occurs in aqueous media. And this explains that some xylose is also produced during the reaction. So reaction conditions have to be carefully selected in order to favor the transglycosylation compared to the hydrolysis. In the ValBran project, we studied the production of xylosides directly from wheat bran that contains xylants. Wheat bran, without any pretreatment, was used as donor, whereas pentanol was used as acceptor of the reaction catalyzed by a xylanase. Reactions were performed at 50 degrees during 84 hours. No co-solvent was added during the reaction. Within these reaction conditions, 520 mg of pentyl beta-D xylosides were produced per gram of xylants present in wheat bran, and the average degree of polymerization of the pentyl xylosides was 1.3. This approach was also tested successfully with longer chain length alcohols such as octanol. We tested the synthesis from two different wheat bran samples, one sample from the BioBased company and another sample from the company ADM. We obtained similar results with the two samples, demonstrating the robustness of our enzymatic process to produce alkyl xylosides from wheat bran. The synthesis reaction was upscaled in a 2-liter reactor in order to produce several grams of pentyl polyxylosides that were provided to ARD company for tests as detergent. We are also interested in the enzymatic synthesis of sugar esters and notably dexylose esters. The synthesis of xylose esters was first developed with xylose and vinyl laurate. The acylation reaction was catalyzed by the lipase N435 from Novozymes in presence of 2-methyl-butan-2-ol as solvent. In contrary to glucose esters, few data from literature were available on 2 xylose esters, and we solved for the first time the structure of xylose esters produced by this enzyme. This enzyme produced 2 monoesters, as you can see on the slide, the 5-oloryl dexylofuranose and the 4-oloryl dexylopyranose. The first one was major, whereas the second was present as trace. The enzymes also produce 2 diesters, the 2,5-D-oloryl dexylofuranose and the 3,5-D-oloryl xylofuranose. Kinetic studies indicated that the major monoesters, labeled 3A on the slide and represented in dark green on the figure, so this major monoester was first produced and thereafter both diesters were synthesized. This demonstrated that the esterification first occurred radioselectively onto the primary hydroxyl groups, leading to the monoester 3A synthesis, and acylation of the secondary hydroxyl groups occurred later on the O2 and O3 positions of the monoester 3A. After 24 hours, the conversion of xylose reached 80%, and 18 mg of total xylose esters per milliliter were produced. In order to study the structure properties relationships of the xylose esters, we synthesized a library of xylose esters as described in the table. The enzymatic acylation was conducted with various acyl donors, leading to the synthesis of xylose ester displaying alkyl chain lengths from 8 carbons to 18 carbons. In case of octanoate esters, the number of alkyl chain lengths was modulated by producing monoesters and diesters. Finally, the degree of polymerization of the hydrophilic part was modulated by producing xylobiols, xylotriols, xylotetraos laurate esters, and a mixture of xyloligosaccharides laurate esters. All these xylose esters were purified and provided to Liège University to evaluate their physicochemical properties as well as their biological properties. And Magali Deleu, in her talk, will present you some results obtained with these xylose esters. Finally, an upscaling of the synthesis of some xylose esters was performed to produce several grams of the most interesting molecules, which were provided to Sopura for antimicrobial tests. As for the synthesis of alkyl glycosides, we investigated the production of sugar esters from wheat bran. In a first step, wheat bran was hydrolyzed with cellulase and hemicellulases in water during 24 hours at 50 degrees. The hydrolysate containing glucose and xylose, and the efficiency of the hydrolysis was comprised between 50 and 100% according to reaction conditions such as the enzyme loading. At the end of the hydrolysis, some wheat bran residues are recovered. Compared to the initial wheat bran, these residues contain lower content of xylans, which are known to display anti-nutritional effect during digestion by animals such as poultry. Furthermore, some cellulase and hemicellulase at the end of the reaction can be absorbed onto the wheat bran residues, giving thus interesting feature for using wheat bran for feed applications, as cellulase and hemicellulase are known to improve the digestibility when added to feed pellets containing lignocellulose. For these reasons, the ValBran partner Inagro is in contact with some companies to test wheat bran residues for feed applications. So after the first step, the hydrolysis of wheat bran in order to produce glucose and xylose, in a second step, the hydrolysate containing both monosaccharides was acylated by the lipase in presence of methyl laurate or lauric acid with 2M2B as solvent and in presence of molecular sieves. The reaction occurred at 50 degrees. In these conditions, a glucose and a xylose laurate monoesters were produced. The glucopyranose was acylated on the O6 position, whereas the xylofuranose was acylated onto the O5 position, as previously described. On this slide, the results obtained during the acylation with lauric acid and methyl laurate at 300 millimolar are presented. For both acyl donors, the main ester produced was the glucose laurate ester, as shown by the blue bars on the graphs. The xylose laurate esters was produced with a lower extent as seen with the red bars. After 72 hours, in presence of lauric acid as acyl donor, the conversion of glucose and xylose was respectively 72 and 13%, whereas the conversion reached 97 and 17% for glucose and xylose respectively in case of methyl laurate used as acyl donor. So higher conversion rates were obtained with methyl laurate used as acyl donor. Finally, we developed an integrated approach for the synthesis of pentyl polyxylosides and laurate glucose and xylose esters. In a first step, a transglycosylation reaction was performed from wheat bran in presence of pentanol as described few slides before. Pentyl beta-D-xylosides were produced with a yield of 520 mg per gram of xylans present in wheat bran. Pentyl xylosides were present in the pentanol phase at the end of the reaction. As indicated previously, transglycosylation reaction is in competition with hydrolysis, and thus some glucose and xylose are also produced during the reaction, as the reaction was conducted in presence of a mixture of xylanase and cellulase. It is important to note that no methyl glucosides were detected at the end of the reaction, indicating that the cellulase did not catalyze transglycosylation reaction. The glucose and the xylose were present in the aqueous phase at the end of the reaction. In a second step, the enzymatic acylation was performed onto glucose and xylose from the aqueous phase in presence of methyl laurate as acyl donor. The two lauryl glucose and xylose monoester were produced at a concentration of 61 millimolar and 10 millimolar respectively. So this slide compares the two approaches that we developed for the synthesis of bio-based surfactants from wheat bran. The separated approach allows producing methyl xylosides and glucose, as well as glucose and xylose lauryl esters from two different wheat bran batches. In the integrated approach, combining transglycosylation and transesterification was successfully developed to produce both methyl xylosides as well as D-glucose and D-xylose lauryl ester from one unique batch of wheat bran. Furthermore, lower cellulase and xylanase loading was used with the second integrated approach compared to the separated approach. So to conclude, efficient enzymatic processes were developed and optimized for the synthesis of alkyl xylosides as well as D-glucose and D-xylose laurate esters from wheat bran. We developed a separated approach as well as an integrated approach from one wheat bran sample. According to the approach, the global yield was comprised between 100 to 150 kilograms of biosurfactants per ton of wheat bran. The data were transferred to ValBiome for LCA and to Vito for TEA. A library of original xylose esters was produced for evaluation of their structural properties. The most promising biosurfactants were produced in larger quantities and are tested by some companies. And finally, wheat bran residues, which were recovered from the enzymatic hydrolysis, are tested for feed application. I would like to thank the colleagues who participated to this work, Professor Plantier-Royon, Dr. Musard, Dr. Rakoto Arivonina, and also Dorian Gérard and Chloé Joquel, who were respectively postdoctorate and technician for this project. And I also would like to thank you for your attention. And now the next talk will be delivered by Magali Deleu from Liège University.
Thank you, Caroline, and good afternoon, everybody. So my name is Magali Deleu, and I work as a researcher at the Jean-Bou Agrobiotech at the University of Liège. So the aim of my talk is to give you an overview of the biophysical-chemical properties of one family of compounds synthesized within the ValBran project. So this family is the xylose esters. So during the ValBran project, different xylose esters with a specific structure have been synthesized in order to investigate the effect of their structure on their biophysical chemical properties, so to contribute to the determination of the structure-function relationships. And as already mentioned by Caroline, there were three distinct categories. So the first one is xylose esters with distinct acyl chain lengths. So we have the C8, C12, C14, and C18 xylose monoesters. The second category was xylose esters with a different number of xylose residue between 1 and 4. So the xylose the xylose ester monoesters with 1 residue, 2 residue, 3 residue, or 4 residue of xylose. And in the third category, we investigate the effect of the number of acid chains by studying the xylose octanoate monoester compared to the xylose octanoate diester. So on all these xylose esters, we explored various biophysicochemical properties. The first one was the potential of the different xylose esters for drug solubilization. The second one is their potential for water decontamination. The third one is their ability as antibacterial agents. The fourth one is the possible application as elicitors of plant defense. And the last one was their potential as detergent agents. So the first property investigated was the potential for drug solubilization. And for this purpose, we measured the iodotrope activity. So what is it, the iodotrope activity? This activity is related to the capacity of a compound to increase the aqueous solubility of poorly soluble solutes. And to evaluate this iodotrope activity, we used a colorimetric assay using the Sudan-1 dye, which will be yellow if it is encapsulated within the iodotrope self-assembly, or orange, dark orange, if it is not encapsulated. So here it's a global view of the results we obtain on all the xylose esters. So as you can see, all the compounds have a relatively low iodotrope activity compared to the reference, the DTAB. Nevertheless, one compound emerged from the others. This is the xylose octanoate monoester. So the xylose esters with one xylose residue and one chain with 8 carbon atoms. So the second property studied was the potential as water decontamination agent. And to characterize this potential, we measured all the displacement capacity of the xylose esters by simply measuring the retraction of an oily film initially formed at an air-water interface in a petri dish. So the global view of the results shows that the best candidate is in red, encircled in red, is the one with 2 xylose residues and an acyl chain with 12 carbon atoms, which is named dixylose laurate monoester. So this is the best candidate for the water decontamination. The next properties was to investigate the potential as an antibacterial agent, and to explore this potential, we measured the capacity of the xylose esters to permeabilize a membrane mimicking the plasma membrane of a bacterium. So first, we formed vesicles with a lipid composition representative of the one found within the plasma membrane of a bacterium, and with a fluorescent probe encapsulated in the internal compartment of this vesicle. So in yellow here on the left of the slide. So after the vesicle formation, we put the vesicles in contact with the xylose esters, and we measure by fluorescence spectroscopy the evolution of the fluorescence intensity. And if the xylose ester is able to form a hole or to destabilize the membrane, an increase of the fluorescence intensity will be observed. And from this fluorescence intensity raw data, we can calculate a percentage of permeability of the membrane. So the results show interesting relationships between the permeability percentage and the structure of the xylose esters. So as you can see here, there is an optimal chain length. The xylose ester with acyl chain comprising 14 carbon atoms have the best activity in terms of permeability of the membrane. So it is the green bar. So in terms of the number of acyl chains, we can observe that the presence of 2 acyl chains gives rise to a higher permeability. And in terms of number of xylose residues, there is no significant effect. So for this activity, so for the permeabilization of a bacterial plasma membrane, the best candidates are the xylose myristate monoester, so the xylose ester with 1 ester residue and 1 acyl chain comprising 14 carbon atoms. And another best candidate is the xylose octanoate diester, so the xylose ester with 2 acid chains comprising 8 carbon atoms. So the next properties was the plant defense eliciting activity, and to first, I have to define what is an elicitor. An elicitor is an exogenous molecule applied on a plant and able to induce the defense response in plants. It's a kind of vaccine for plants. So how we did this measure, we know that from other studies that the eliciting activity can be related to the ability of the compound to perturb the organization of the plasma membrane of the plant cell. So to evaluate this membrane perturbation, we performed similar experiments as we did for the evaluation of the antibacterial potential, except that in this case, the lipid composition of the vesicles was representative of the plant plasma membrane. And we would have expected to see a high activity of the xylose esters, as we know from other studies that other glycolipids like rhamnolipids have a very high eliciting activity, but unfortunately there was no best candidate for this property. There was no activity at all. So the last property investigated within the ValBran project was the detergent property. And to evaluate this property, we measure 2 parameters: the adsorption of the xylose esters at an air-water interface and their aggregation behavior by a tensiometry technique. And here are the results. We observed that the middle chain compound, so comprising 12 or 14 carbon atoms, developed the best surface active properties. That the presence of 2 acyl chains is better than 1. And that a limited number of xylose residues, 1 or 2, gives rise to a higher surface activity. So, in terms of detergent activity, the best candidate is the xylose octanoate diesters, as it shows the lowest critical micellar concentration and the lowest surface tension. To further investigate the effect of the molecule structure on the surface activity, we used molecular modeling to determine the three-dimensional structure, the conformation of the xylose esters at an interface. And I illustrate this part by one example, the xylose octanoate diesters. So we started from the 2D structure on the left of the xylose octanoate diesters and performed an energy minimization calculation to determine the most favorable conformation and orientation of this compound and at a hydrophobic hydrophilic interface. And as you can see, the polar head group in green is protruded in the hydrophilic region of the interface, and most of the acyl chain in yellow is in the hydrophobic side, and the molecule globally adopts a vertical position at the interface. And by analyzing the molecular modeling data from all the molecules, from all the xylose esters, we concluded that the hydrophilic balance of the xylose esters and their orientation at the interface, in other words, the optimal occupation of the interface by the compound, are the most important parameters playing on the surface activity of these compounds. So this is the end of my talk. So to finish this talk, I give you a summary of the structure function relationships that we have highlighted. So for the drug solubilization, it is the compound with the lower chain length which gives the best activity. For the water decontamination, it is a compound with 2 xylose residues. For the antibacterial activity, 2 structural parameters are playing: the acyl chain length and the number of acyl chains, with the best results obtained for 2 chains with 8 carbon atoms or 1 chain with 14 carbon atoms. For the plant defense elicitation, there was no significative activity. And for the detergent property, again, a double acyl chain gives rise to the best surface activity. So thank you for your attention, and I would like also to thank my colleague Catherine Kemotti and Richard Plantier-Orion for the technical help and for the design of the synthesis of the xylose esters. And it will be a pleasure to answer to your question at the end of this session. And now I give the floor to Winnie de Jong from Vito. Thank you.
Winnie, your microphone is off.
Okay, sorry. Thank you very much. Okay, so thank you, Magali. And so good afternoon, everybody. So in what follows, I would like to give you an overview of the results that we have obtained on the enzymatic synthesis, but also the techno-economical analysis of the production of glucose and xylose laurate esters at bigger scale using higher substrate concentrations and membrane-assisted solvent recovery. So in our part of the research, we try to find out how we can bring these sugar ester synthesis routes that are developed in the ValBran project closer to the industrial level. And to obtain that, we wanted to answer 4 questions. So first of all, we wanted to know which acyl donor should we use at industrial level. We also wanted to know, can we increase the product titers by increasing the substrate concentration? Can we increase the amount of product that is produced per reaction run by increasing the reaction volume? And can we use a techno-economical analysis to determine factors influencing the efficiency of the processes? Before I continue, I would like to stress that all results that will be presented were performed using pure xylose and glucose, so we did not use any wheat bran in our experiments. So as you might know, sugar esters can be produced by using the esterification or the trans-esterification reaction. So in the esterification reaction, lauric acid is being used as a donor, as an acyl donor, and water is being produced. While in the trans-esterification reaction, vinyl esters or methyl esters are used as acyl donors. In the case of vinyl esters, vinyl alcohol is being produced, and this product tautomerizes to produce acetaldehyde. When methyl esters are used as an acyl donor, methanol is being produced. However, in ValBran, we wanted to produce products that are mainly used in the cosmetic and the feed industry. So therefore, the use of vinyl esters or methyl esters to produce these sugar esters is less appropriate because these acetaldehyde and methanol can form a contaminant to these sugar esters and cannot be used in feed and cosmetic applications. So therefore we asked ourselves, can we also use lauric acid as a donor? Because then is water is being produced as a byproduct, which is less harmful. But indeed, and when you take a look to literature, most of the time these trans-esterification reactions are being studied, and there is not a lot of knowledge known about the esterification reaction. So to find out which of these acyl donors is the best, we performed the following experiments. So xylose was mixed in equimolar amounts with the 3 different acyl donors, and all substrates were present at a concentration of 40 millimolar. We used lipozyme 435 as a lipase enzyme, and we added different types of molecular sieves that would remove the side products water, acetaldehyde, or methanol to pull the reaction to the product side. We performed these reactions in the green solvent 2-methyl-2-butanol because then we get a better solubility of the hydrophilic sugars, but also the hydrophobic acyl donors. And the reactions were all performed at 200 gram scale at 40 degrees. So when we take a look at the substrate concentration in function of time, then we can see that the conversion in the presence of methyl laurate is a bit higher than the conversion in the presence of lauric acid or vinyl laurate. However, we can also see that in the case when methyl laurate or vinyl laurate are used as a donor, these donors are first hydrolyzed into lauric acid, and in fact, it is the lauric acid that is being used mainly as the acyl donor. So if we take a look to the right side graph with vinyl laurate, you can see that after a reaction time of 3 hours, there is no vinyl laurate left anymore in the reaction. So the blue bar has converted to the gray bar, which means that the big part of the vinyl laurate has been converted to lauric acid, and then these gray bars decrease in height in function of time. So the lauric acid is being used as the acyl donor. So based on these results, we concluded that it's best to use, the lauric acid as the acyl donor since it is the cheapest compound, but also it produces water which is less toxic than the acetaldehyde and the methanol that are produced in the case of methyl and vinyl. The type of acyl donor doesn't have an effect on the product that is being produced, since for all three acyl donors, both monoesters and diesters are being produced, as you can see on the right side on the TLC profile. So from these first tests that were performed at very low concentrations of 40 millimolar of substrates, we got a product titer of 10 gram of product per kilogram of reaction medium. So the next question we asked ourselves is, can we increase this titer by using higher substrate concentrations? So therefore, again, a reaction was performed between xylose and lauric acid, and we used a concentration of both substrates of 100, 500 or 800 millimolar. The reaction conditions are the same as in the previous slide, except that for these reactions at higher concentrations, we used a temperature of 60 degrees, since then the solubility of the sugar can be increased. So as you can see on this slide, the conversions of both the xylose and the lauric acid are the same for the three used concentrations of substrate. However, as the absolute concentration of the substrate in the 800 millimolar substrate condition is higher than, for example, in the one of the 100 millimolar substrate, higher product titers could be obtained. So indeed, in assuming that the product contains 18% of monoester and 20% of diester, a product titer of 30 gram per kilogram of reaction medium could be obtained when 100 millimolar of substrate is being used, while we could increase the product titer to 200 gram per kilogram of reaction medium by using the higher substrate concentration of 800 millimolar. We could indeed see by TLC that monoesters and diesters are produced for when the different concentrations of substrate were used. However, it was not possible to really quantify these products because no analytical standards are available. So that's why we assumed that the product contains 80% of monoester and 20% of diester, but these values should be verified in the future. From these products, these xylosolaurate esters that were produced, we could purify them from the reaction medium using a purification method that was developed by the University of Reims. And we could see by TLC that all of these purified products are a mix of mono and diesters. The xylose content is similar in all of them, so it doesn't matter which concentration of substrate you use. The xylose content after purification is the same, but they contain a different amount of lauric acid. So the lauric acid content will increase when the substrate concentration increases. And these samples that were produced at different concentrations of substrates have been given to determine their antimicrobial activity. So next to xylosolaurate esters, we also studied the production or the synthesis of glucosolaurate esters, and the same conclusions as for the xylosolaurate esters can be taken. So first of all, methyl laurate and vinyl laurate are hydrolyzed also in these reactions to lauric acid. So lauric acid is again the best acyl donor, and similar acyl donor and glucose conversions as for xylose are obtained. What we could see when at the right side, so the right graph indicates that when the substrate concentration is increased from 40 to 100, 500 and 800 millimolar, the percent of diesters increases. So for higher substrate concentrations, higher amounts of diesters are obtained, and so lower amounts of monoester. And for this case, with the glucosolaurate esters, we could really quantify these esters because together with the University of Reims, we were able to produce or to purify some analytical standards. Again, approximately the same product titers as for xylose esters are obtained, ranging from 8 gram per kilogram reaction medium for 40 millimolar of substrate concentration, going to 176 for 800 millimolar substrate concentration. For these glucose laurate esters, we also wanted to know if we can increase the amount of product that can be produced per reaction by increasing the volume of the reaction medium. So sometimes when you increase the reaction volume, the conversions will go down. So to test this, we worked with glucose and lauric acid at 40 millimolar, and we produced these the esters using a reaction volume of 200, 400, and 800 grams. And as you can see in the top file, the lauric acid conversion decreased a little bit when the reaction volume increased. And we could also see in the bottom graph that when the reaction volume increased, we have a bit more diester than at 200 gram volume. So probably due to the mixing of the change in mixing in these bigger volumes, we get a bit lower conversions, but it is not a dramatic decrease in conversions. And we could then also determine by increasing these volumes, reaction volumes, that we could indeed in one run obtain more product. So although these reactions are already working quite well, we wanted to further optimize the efficiency of these processes, and we wanted also to compare the price of the products that we make with our process to the price of the products that are available on the market. And to study these questions, we used a techno-economical assessment. So this techno-economical assessment comprises 4 steps. First of all, there is the market study. So in this step, we will determine the characteristics of the product that we want to make, but also we will screen the market to look for the market price. Then we have to develop a process flow. So in this process flow, the different steps that are taken in this process are elaborated, but also the mass and the energy balances are made. Of course, in the economic analysis, the price of the product will be determined by taking these mass and energy balances into account, but also the OPEX and the CAPEX costs. And finally, we will do an uncertainty analysis to see which parameters of the process determine the process, but also the price of the product. And then it is the aim that that specific parameter can be optimized to optimize the process and to also decrease the price of the product. So in ValBran, we performed a quite simple techno-economical assessment since we only took the mass balances into account. So by determining the amount of substrates, but also enzymes and substrates and molecular sieves that are used to produce 1 kilogram of product under the same reaction conditions, and by taking the price of these different reaction compounds into account, we could determine the product, the price of the esters that are produced in ValBran. So I have to stress that in this data, so no CAPEX and OPEX costs are taken into account. So it's only the cost of the reaction compounds that are taken into account. So based on the amount of product that is produced when we react 100 millimolar of xylose with 5% of lipase and 10% of molecular sieves, we know how much solvent, substrate, enzyme, and molecular sieve have to be used. And based on the costs of these different reaction compounds, we could determine that when we use 100 millimolar of xylose, that we come to a product price as high as 2,000 euros per kilogram. However, when we go to a substrate concentration of 800 millimolar of xylose and convert these 800 millimolar of xylose under the same reaction conditions as used for the 100 millimolar of xylose, the amount of solvent, but also enzyme and molecular sieves will decrease because the process will become more efficient. And based on that, by using 800 millimolar of xylose, the cost of the esters will drop or will decrease to 300 euros per kilogram of product. However, we also know from by a study performed by the IARPOL in France that the selling price of these esters is between 3 and 4 euros per kilogram of product. So we are still far away. To go closer to these selling price, it is indeed needed to recycle the solvent, to recycle the enzyme, and even to recycle the molecular sieves. So by recycling the solvent for 70%, the enzyme for 90%, and the molecular sieves for 100%, we could decrease the price of the ester with a factor of 10 at 800 millimolar of xylose. So the price decreased from 300 euros to 30 euros. Since we are still, let's say, we are closer to the 3 to 4 kilogram, to the 3 to 4 euros per kilogram, but we don't reach it yet. So therefore, further optimization of the reaction conditions are needed. And by using a solver analysis, we found out that to reach this production cost of 3 euros per kilogram, we have to decrease the lipase concentration that we use in our reaction from 5 weight% to 0.3 weight%. In addition, the xylose conversion as well as the lauric acid conversion has to further increase, and also the solvent has not to be recycled for 70% but for 88%. But as already said in the beginning of the presentation, in our calculations we only take the mass balance into account, so we don't take the OPEX and the CAPEX into account. So to give a bit of playground, let's say, for these OPEX and decrease the product 20 euros to get this lower price. We know that we have to decrease the amount of enzyme even more from 0.3% to 0.1%. So it was very clear from this data that the most important parameters that determine the cost of the product are the enzyme, then the recycling of the solvent, and the recycling of the molecular sieves. And in this last part, I would like to introduce to you that we tried to recycle the solvent by nanofiltration. So we used nanofiltration to selectively remove the solvent from a reaction mixture that contained the solvent together with the lauric acid and with glucose mono and glucose dilaurate. And as you can see here in the last slide, there is one polymeric membrane that retains all the substrates and all the products, but that leaves very selectively through the solvent. So that membrane is a candidate to be used for solvent recycling in this process. So finally, I would like to end with some conclusions. So xylose and glucosylate esters can be produced at a substrate concentration from 40 to 800 millimolar. They result in a product titer ranging from 8 to 200 grams of product per kilogram of reaction medium, and we could increase the reaction volume from 200 to 800 grams. So the main conclusions or the data are that the production costs based on only the reactant consumptions is still much higher than the targeted selling price of 3 to 4 euros per kilogram of product. But the process requires further optimization to increase the substrate conversion, decrease the dose of enzyme, which is a very important factor, and ensure the recyclability of the solvent and the enzyme. And to recycle the solvent, it was possible to use a nanofiltration polymer with a very good retention of the substrates and the product, and with the release of the solvent. So finally, I would like to thank my colleagues who generated all these very nice results. So Marta, Lieve, and Yamini. I would like to thank the ValBran team for the nice collaboration, and I would like to thank you for your attention. Thank you very much.
Thanks, Winnie, Caroline, and Magali for this nice presentation on the main outcomes of the ValBran projects. As you have shown, that you have developed different alkyl xyloside and different, I would say, esters and sugar esters with antimicrobial or with detergency properties. You have identified different candidates for all the application which has been targeted, and you have made, I would say, technical analysis showing that it's perhaps possible with more optimization to target selling price from 3 to 4 euro per kilo. So we can take now some questions, technical question, and also a question on industrial barrier to the markets. The first one will certainly go to and speak about the optimization of the process and the next steps. You mentioned that it's very important to recycle the enzymes or to reuse the enzyme. Can you think also about the immobilization of the enzyme, and what do we know about the recycling of the lipids today? Is it easy to recycle these lipids?
Yeah, I suppose it's a question for me.
Yeah.
Yes. Okay. So the lipozyme 435 is, let's say, the well-known Novozyme 435. Yeah. So it's already immobilized. So yeah, and we just buy it, let's say, from Novozymes. Yeah, recyclability of these lipase is yeah, possible because we already did it in other projects. The easiest way to recycle or to reuse, let's say, the enzyme is to use it in a column. But as here, the solubility of the reactants, so the sugar and the fatty acids, isn't that high, the use of a column to reuse the enzyme will not be easy. So indeed, we should think like, how can we recover the enzyme, the immobilized enzyme from the reactor and then reuse it? But that will also be a challenge.
Okay. Yeah. And you can immobilize it or not?
No, it's so the lipase is immobilized. You buy it on an acrylic bead.
Okay.
Yes.
And can you reduce the amount of the lipids?
The fatty acids, or what do you mean?
The lipase.
Yeah, the lipase. Yeah, indeed. That's indeed another approach that we have to use. So now we used 5 weight %. We also know that 1 weight % is possible because that was the concentration that is used by Urca in their experiments. But we indeed should go lower. Yeah.
Okay. I would like to have some information regarding what has been surprising for you when you have get the results on the properties and the Russian properties studies. And the question is for Magali. What has been surprising for you?
Sorry, I didn't hear the.
Yeah, when you
question.
get the results
yeah.
On the Russian properties from the different sugar esters, what's surprising you on the applications?
What's surprising? What is surprising?
Yeah.
What is the best results? According to me, the best application of this sugar ester is as a detergent agent. So they showed interesting surface activity, good surface activity compared to other glycolipids. So according to me, this is the best.
Okay, the best candidate is the detergency. Okay.
Yes.
And the other one?
The other one, the alkyl.
Yeah.
I don't know exactly because we didn't do all these studies on the other family of compounds synthesized during the ValBran project. So I cannot answer to this question now. Okay.
And a question regarding the next step. So you have different samples, I think. So you will certainly send the sample to different industrial. And but what could be the next, I would say, projects in order to do scaling up and to optimize the synthesis of the different biobased surfactants?
I can answer.
Yeah.
We discuss this between the partners, and we think that if there will be a next project, it will be focused onto the scaling up indeed, because we developed our processes at laboratory scale, and so Vito upscale the esterification reaction, but we did not test this upscaling for all the enzymatic reactions. So we really need to upscale the reactions in order to have interesting data for LCA and TEA. And another aspect will be to study the formulation of our molecules, because we didn't manage this part within our project. We produce molecules, we test the properties of the molecules, but we didn't test the characterization of the molecules into different formulations for application. So it will be also an interesting step for a future project.
Okay.
But my colleague can add information, of course.
Yeah. There is some questions regarding, can we also look at continuous process, for example, the use on enzyme in identified process? And
yes, okay, it was the idea to study it, but indeed we bumped in a bit on the solubility of these substrates. So we are a bit puzzled over there, like what will be the best approach. Maybe with the alkyl polyglycosides, we were thinking about the enzyme membrane contactor maybe to apply. But yeah, okay, we didn't come that far in the project, but it could be an approach, but it has to be studied.
Okay. And a question which is more for industrial, I would say, what about the rich situation of the biobased surfactants which has been produced? There is already a product, I would say, a product which is known and approved or not? And
perhaps Boris, about alkyl polypentosides, you have information about REACH?
I think, Boris, are you there? Okay, it doesn't matter, but it's a question we can ask after the. So next, last questions that we have worked doing 4 years between, I would say, Flemish, Belgian, and French players. What for you the advantage of working together through an Interreg project?
Perhaps, Jackie, I prepared a slide for this,
yeah.
So perhaps we can go through the presentation if you, I don't know, where is it? Oh, it is before, I think.
Okay.
Yes, it is this slide. Is it okay if I use it?
Yeah.
So yes, so as cross-border added value of this Interreg project, this cross-border added value comes mainly from the complementary skills and approaches that were developed by the 9 partners of the project. So you can see on the slides that different skills were present into the project. So some biocatalysis skills, bioprocess engineering, separation and characterization of the molecules, properties of the molecules, LCA and TEA analysis. So today we decided to present you some results obtained during the ValBran project, but numerous data were obtained, notably for the LCA, but some are still under validation. So we selected an overview of the main important results that were obtained, but you can see that the collaboration between the partners was really strong, with arrows that are indicated. And also another important cross-border added value of the project was the large networks from Valbiome, IAR, Inagro, Greenwind, and Catalysti. And this allowed to improve our communication actions during the project, because at the end of the event, we will briefly present the highlights about the communication actions. So it was also a really important part of the project to communicate to companies, to society during different events. So the cross-border added value was there.
Okay. And do you think now you well known, I will say, the constraints, the industrial constraints of the biobased surfactants, and thanks to the projects?
Yeah, yes, the interactions between companies and the partners were important during the project in order to obtain information on which molecules are the best one for companies. So yes, we improve our knowledge about this, but we still need information from companies as academic scientists.
What type of information? Do you know, for example, what is the behavior of this type of biobased surfactants in formulations and cosmetic formulation? What do you know exactly on the really when you blend it with different others, with different pigments, with other substances, what's happened?
In the project, we didn't take care about this information, so we don't have this information, but that is an important part, as I mentioned previously, for a future project. We want to know the loading of these molecules to add in the different formulations, the compatibility with other additives and other molecules. But we didn't study this during the ValBran project, but now it is very important to learn about this.
Okay, and regarding the toxicity and ecotoxicity, have you some data which on the top candidate, I would say, you have mentioned?
What I know from ARD, which produce alkyl polypentoxide, is that there is no toxicity. So we can suppose that the molecules that were produced during the project also don't present any toxicity and ecotoxicity, but it will have to be proven. Yes, we have to test this, but it's supposed to be not toxic. Okay,
so it will be certainly the next steps, but if you want really to make the scale up and to really look at the industrialization, that should be something which should be very important. So what I propose is now to make a break from one quarters, and we will come back in 1 quarter with 2 companies, Sopura and ARD, for speaking about the scaling up, but for speaking about the industrialization of the biobased surfactants. Especially ARD will I will say develop and compare the properties from alkyl polypentoxide and alkyl polyglycoside, and also Sopura will show different applications for I will say for the detergents, for bottle, for air wings, and so on. Welcome back to the second session of the ValBran projects, which is dealing with biobased surfactants coming from ValBran. We are more speaking in this session about the industrialization of the biobased surfactants, speaking more about the positioning of the biobased surfactants, the markets and different applications, and also I will say the barrier to markets and the barrier to the industrialization. And one of the presentation from Sopura is called From Dream to Reality. We have in the first part look at what we can eventually do without a lot of economical constraints. Now we are more speaking about what are the market constraints and market barriers. So I will first of all give the floor to Mr. Verschav from the Sopura company, which will give us some example of different application of biobased surfactants, and also
which will speak about the constraints as is a regulatory, I would say, expert in the area. Okay, the floor is yours.
Okay, thank you very much, Chairman. Good afternoon, everybody. Yeah, the title is indeed Green Sustainable Chemistry, but it's a dream or reality, so that I would like to highlight. It's an important thing. So, one second, please. Yeah. My exposure, in fact, we have only 15 minutes, so I will try to limit myself to the most important things, because 15 minutes, that's quite quick. So a small introduction about the company Supura. Some views about green sustainable chemistry. How is the market looking to this? Some specific examples to illustrate, in fact, when we use surfactants and what kind of businesses and applications are we confronted to the use of, let's say, of surfactants. And then some reflections, yes, is it all a dream or reality? First of all, perhaps the company Supura. Supura is a company, a global company, meaning we are present in more or less 70, 80 countries worldwide. And we are selling cleaning, sanitizing, and water management solutions. Why I'm mentioning a global company? Because, of course, if we develop product formulations, we need to take into account from the starting point in our development that we will sell those solutions in 70, 80 countries, meaning that from time to time for corporate organizations, if we bring a solution to the market, we have from the beginning and from the starting already in this development process to take into account all the different constraints that we are confronted to, because otherwise we fail, in fact, in our developments. And it is more and more important that we do this. I mentioned by doing this, we do this in a responsible way to reduce, of course, all environmental impacts of, within our activities, with high quality standards to service our customers, and of course, trying to leverage the proven technologies and formulations all over the world and translate it in what we call best practices. Why did I put this first slide? It's because we are more and more confronted to not only the selling of, let's say, cleaning, sanitizing, and water management solutions, but the industry, the end users of our business solutions, they put higher requirements than only, let's say, product formulations to us. They ask that a solution that we bring to them reduces energy, reduces water consumption, reduces the impact on the wastewater, etc., etc. So if we offer a solution, and I heard this in the previous presentations, we don't know exactly all the details about the constraints of the industry. You have to take this also into account, I would say, in the development of surfactants from the beginning, because there are regions from time to time in the world which can't use phosphor-based molecules. There are regions in which we know that upfront, the development, that if we bring a solution to them, we have to take into account that the water which is drained to the wastewater, that it will be reused and even from time to time used by farmers. So there are a lot of constraints that you have to know upfront if you do development or research on molecules. The use of surfactants in our business, you're all very intelligent people, so you know this, but we are using, in fact, anionic surfactants, cationic surfactants, amphoteric surfactants, and non-ionic surfactants. And we use them for, let's say, in a nutshell, 5 functionalities. I forgot one, but I will mention it at the end. It's just wetting agents, solubilization agents, dispergent agents, emulsifying agents, foaming, defoaming agents, and the last one is supporting molecules which can help also to support biocidal activities. It was mentioned by Caroline in her presentation. We used one of the molecules which was mentioned this afternoon also for a biocidal product formulation. So the question, which is the title of my presentation, is green sustainability chemistry, is this a dream or reality? And what in fact are the market expectations? Because we are talking about green chemistry and about sustainability, but at the end, yeah, what is possible, what is realistic, and what is a dream, in fact? And that's why I put the next slide, something which is very recent that you have to know, and which gives already, I would say, one part of the answer to the question if this is still a dream or reality. I think 3 weeks ago, the Commission published what they called the Chemical Strategy for Sustainability. So it's a very intensive document which still have to be submitted to the European Parliament and validated by the European Parliament and also some, let's say, stakeholders. But the document is very important because it's a kind of documents which says in fact that for the next 20, 30, 40 years, Europe should evaluate, should move towards, let's say, a green society. And it's the first time that this is said in, let's say, in such explicit wordings. So to pick up the question, is this a dream or a reality? For me, it becomes reality. Because they would like to translate this document into new regulations which should support more, in fact, the move towards those sustainable green solutions. And for us, this is an important sign which have been given to the industry. It could be at the same time be a constraint because also the Commission, they are aware of the fact that you can only bring solutions to the market if the regulations moves at the same time. And if you don't do this, perhaps the cost will remain too high. The industry will never take the solutions which are proposed because they are looking for the cheapest solutions, perhaps with the highest impact on the environment and energy cost. So that's for us a very important statement and document that they published. So another point, in fact, so this was my view on how is the regulations, in fact, looking to this. Now, a small reflection regarding how is the end consumer looking to this. And that's what I would like to do here on this slide is to highlight something which is very critical and important for us. You have to distinguish different kind of end users. If you look to the cosmetic industry, and you're aware of this because you proposed already, and I would say you did quite a good job on surfactants which can be used in the cosmetic industry. The cosmetic industry, you see that there is a boom towards, let's say, things which are sold with claims that this is bio-based. And the market is moving towards those products because there is a very good structured communication supported by a regulation which is behind. So if we sell hygiene solutions to the market, you still need to be aware of the fact that we are suppliers of formulations to the B2B industry. It's written on the document C2C, it should be B2B industry because we are selling, in our case, let's say 80% of our customers is the beverage market, is the beer market, beverages, soft drink market. So we are providing hygiene solutions, but I would say, with a question, did you ever wonder if you drink a Coca-Cola or if you drink a beer, were you influenced in your choice of your drink if the hygiene solution which is proposed to clean all the vessels or the bottling machines, if this is done with sustainable or not sustainable solutions. I don't think so. I don't think so. If you're in the cosmetic industry, you buy something and you're influenced by the packaging because it's mentioned, it's based on bio-based substances. And this is not a detail, but this is a very important fact. Why I'm claiming this? Because as long as the regulator don't support more our industry by making the link between what the end consumer is consuming and paying for and what we are providing as hygienic solutions, which are more greener and sustainable and bio-based, the process will not happen. So, and I would say, if the end consumer is motivated for buying something based on a claim and it is written in the communication, I think, and we think that he will be ready for paying for this, but also the industry who will use our products will be ready for using them because the consumption will be and the sales process by his industry will be impacted by this. And this is still today a very important link which is missing. The link between what we are providing and what the end consumer finally is using. This is different for some industry, like I mentioned, cosmetic industry, laundry industry. If we have to use products in our housekeeping, I would say, we are today influenced by the greener aspects. Why? Because if you have to put something in your washing machine to clean clothes, you will look to the packaging. You will choose solutions which are perhaps more sustainable. So in some areas, customer is impacted. In some areas, he isn't impacted. And this is what I write down here in the document at the right. Between product and consumer, there are several, in fact, we call it regulatory positionings, which are talking about impact on ecology, sustainability, etc., etc. But as long as the claim which is sent to the consumer is not taken into account in the whole chain, the move will be theoretical, but the move will not take immediately. So as you can see on this slide, authorities helped already supporting this process by putting in place eco-labels, by putting in place, I would say, EC norms, standards to support a bio claim or to support the life cycle, etc., etc. But all this for us is still a puzzle because in reality, in the B2B business, it doesn't really help you. If you look to what's behind today the eco label, you can claim the use of formaldehyde with an eco label. Is this sustainable? Not at all. So it's still, so those claims are still a little bit artificial. There have been temptations in the market by stakeholders to help the industry to move towards those better claims. But for us, being in the B2B business, it's not sufficient. And as long as this process doesn't take place in a structured way, it will be still a little bit more dream than reality. I would like now to give some examples of where surfactants with those 5 different functional roles in our industry are extremely important and interesting. One typical example, but I think that I missed with my slides because I have first, no, it's correct. I go back. Yeah, you all know beer filtration or filtration of liquids. It's done more and more today with membrane filters for sustainability reasons. I've been working for 20 years in the brewing industry, and when I started my career, we worked with those traditional plate filters, which were working with Kieselguhr to obtain your final filtration of beer. So after the fermentation, maturation, you need to filter your beer. So these technologies have been replaced the last years by membrane filters because Kieselguhr is not at all sustainable. So, but one of the big problems with those membrane filters is that you're using membranes, and to have a membrane filtration, that's a tangential filtration, you have to manage what we call the transmembrane pressure. And so transmembrane pressure evolution is impacted by the presence of solids, the presence of complex molecules that you have to filter, in fact, on your membrane filter, and it makes that after a while your filter is completely blocked. So you have to eliminate complex polysaccharides, you have to eliminate complex polyphenols, tannins, etc., etc. For doing this, the use of specific surfactants is very interesting because it helps you to avoid the blocking of those filters. It helps you to clean your filters in a much more sustainable way. So that's a very typical example which demonstrates the use of surfactants. And at the end, I would say, surfactants help enormously for the end user, and that's what we are aiming for, to reduce the total cost of ownership on his filter, because you have a reduced downtime of your filter. You can more quickly eliminate difficult particles which are responsible for the fouling of your filter. You increase automatically the capacity of your filter. You reduce the global cost per hectoliter beer which is filtered, and you have also at the end a lower dependency on the variability of your raw materials. So here, in fact, the cost of your surfactants should be evaluated with all the consequences that the use of this surfactant has on the practical application in the field. So this is one. We are worldwide providing solutions for those membrane filters, but this is one, for instance, in which we know that the cost per kilogram of this surfactant is high, is very high, but at the end, if the brewer makes his calculation, for him, the global cost is reduced. We call it the total cost of ownership. Another one, which is a very recent one also, what I would like to illustrate is the validation of the European Commission, which happened and took place, and we are proud about it as a Belgian, let's say, company, is the validation of the product Sopoclean NR at the European level. It's the first European non-rinsing detergent disinfectant which is allowed by the European Commission. We can sell it since the beginning of April in all the 27 countries in Europe, covered for a period of 10 years. So it's based on a European authorization. This product was completely developed on natural substances. So the active ingredients are octanoic and decanoic fatty acids, which are extracted from palm oil. But it reveals an evolution in which we believe as a company, if you can move towards sustainable disinfectants, which make that at the end, if you have some residues which remain on the surface, but for the end user of this product, this is great, because if he has to disinfect a surface or a vessel, and he knows that the residues which are present should not be rinsed off, and that even by having them in your end liquid, food or beverage, they are okay for Europe, because it could be that they are naturally already present, and then it's not a matter anymore about cost and a discussion about cost. You have to take the use of those formulation. You have to look to this in another way what is a global, let's say, impact and what is the global advantage of the end user of your formulation. So we recently, and we still believe in this project, we have used one of the molecules which have been shown this afternoon, and we are now evaluating if it helped to boost the biocidal activity, because it's not because we are using already validated octanoic and decanoic as active ingredients, but is if additional natural substances can create an additional impact, it will help us to obtain a better solution at the end. So this is something which is very interesting for us, and it demonstrates again, the potential of having those bio-based solutions available. The next one is also a very interesting one. We are the leader in the world and solutions that we provide to the market for the cleaning of returnable bottles worldwide. So, but for cleaning bottles and returnable bottles, for an end consumer, this is like, he's not thinking about this. But if you have to clean off bottles in 10 minutes, 80,000 bottles per, let's say, per hour, and you know that those bottles were in the market, 70, 80 countries, where you don't know for which purposes they have used after the emptying of the bottle, the bottle, with different glues which have been used to label those bottles, with different kind of crown corks, etc., etc. But the process of cleaning in a correct way a bottle is really a headache for us, and it's market linked. So we use a very specific surfactants for cleaning those bottles, and we have to safeguard, of course, product integrity with those surfactants, because big groups like Coca-Cola, like Pepsi Colas, like the Heinekens and the Carlsbergs, if they would find back in their bottle, let's say, one residue in terms of ppms of something which has not been rinsed off during the cleaning of those bottles, you really have issues with those companies, because product integrity is a very, very important thing for them. So also here, I would say bio-based surfactants are very interesting, but it's important that they are competitive in the market, because it's a competitive world in which we work. So you have to be aware of this. It has to be price competitive, and it could be an opportunity at the same time, because you have to know, to clean off bottles, people are doing this with high volumes, and it's millions of kilograms of caustic which is used. It's energy, high energy consumptions which are involved in this, because bottles are cleaned off at the 75, 80 degrees Celsius for 20-25 minutes. So all solutions which could help to bring better chemistry, greener chemistry to clean off bottles, making, let's say, the process possible to reduce caustic concentration, to reduce energy consumptions, that would be great. So it's just an example to illustrate again that the offering of new surfactants to the market should be also from the beginning of be put in the picture of, can this surfactant generate a global reduction in total cost of ownership for the end user, water consumption, energy consumption, etc., etc. So it's more than just making the overview of it costs X euros per kilogram. It goes further. The next one, in which we are involved with some research center all over Europe, is a very important one, something which was really atypical for our industry, but We have been asked by a research center, and we signed off this agreement for this, if we could bring a solution to the market to better clean the wings of the planes. So it's a project which is called Stellar, Clean Sky, which is financed by Europe. And there is a development of a specific angle on the wing of a plane, which makes that if you have insects, which, during, let's say, the flying off of a plane, insects or forming a kind of deposit on a plane, and it impacts for more than 5 to 10% the consumption of kerosene. So CO2 emission, which is incredibly high, only because some micrometers of insects are on the wing of a plane. So this have been studied by the big companies which are active in this industry. So when we have to work back, we have to provide solutions to clean off, in fact, those insects. But at the same time, we have to think about the environmental impact of our cleaning solution, because in a lot of airports, they do not accept anymore that they have bad chemistry, let's say, which is spilled all over the, how do you call it, the airport. It needs to be sustainable chemistry. So that's also one of the typical projects financed by Europe in which we are involved. Which can be door openers for for new kind of surfactants. And so very interesting. So as a conclusion, I would say we think that the green chemistry and sustainable solutions bringing to the market, yes, we do believe as a company that this is the way to go. And you have seen it, it's supported by this new statement written by the European Commission. But at the same time, I mentioned that you have to look to the market in which you use this. If you look to the market, to the B2C market, I would say this is okay for cosmetics, this is okay for laundry, because the end consumer sees directly the use of this chemistry and can translate this in a consumer message. For the B2B2 market, there is still, in fact, some support which is required by the regulator. And I think that the regulator should install stimuli. This could be tax stimuli. This could be new regulations in terms of the way that you have to communicate, but to combine, in fact, the sustainable hygiene solutions with the end user awareness of this. Fourthly, I think that green chemistry, but that's a door opener, I think, which you all know. It should be, as much as possible, be better than actual solutions. If you can bring an added value compared to what is already be on the market, which is already present on the market, it's better, of course. It's better that it performs better than existing solutions. And I mentioned it already, it should be economically viable, because we are in a very competitive market. I didn't mention it in the beginning, but we have been bought by a French company, Cursia, since last week. So they have been a big announce, and we are proud about it. Cursia is a French group. So we are merged now with them, but it has to be validated by the European Commission. So this should happen for the end of the month. But also this company believes in sustainable chemistry. But the same thing, it has to be economically realistic. So and that's an important thing. And I mentioned also a last reflection. And I think that, Caroline, you mentioned it in a discussion with Jackie. I think that the industry and the research center should sit together more upfront the starting of a project. Because I'm even seeing this every day in a relationship with my commercial people. If you develop and you don't bring into account starting all the constraints that you will be confronted to, markets, and also from the regulatory point of view, it could be that after 3-4 of work, years of work, that suddenly you're aware of something which, has not been exactly, looked into depth. So to conclude, I would say we believe in the dream, but if the dream should become 100% reality, there are still some important steps to be taken. So thank you.
Okay, thanks, Willy, for your presentation, Stefan. And I think we are sharing the same thing that we need, especially at low TRL, to have the investment on different companies in order to really orientate the research and taking into consideration the constraints. And in the bioeconomic clusters, we have developed a guideline for the scale-up of bio-based product, and it's very interesting to take care about this. I will have some questions regarding Sopura activities, and you didn't mention, but which quantity of surfactants you are using today for the different application, especially for the biocide, for example, you put on the light.
So I can't say it in tonnages because I would reveal
The percentage, if you cannot mention the quantity, but the percentage, is it 1% or 10%?
If you think about a biocide product, you have to take into account between 1 and 3%. If you think about solutions for instance, bottle washing, this could be higher, and this should be higher. So it really depends on the kind of application that you're confronted to. But I would say if you think about a biocide product, 1 or 2% is a good indicator. Yeah.
Okay. Regarding, we say that it's important that companies come to the project and the collaboration with the academic. I would like to know what are the tests you are making on the different samples in order to validate it for the scaling up, for example, which will be as a necessary, I will say, required specification for going to the pilot scale, for example, now.
Yeah. What we are applying, and that's, I would say, it's upfront. If we would put a claim on a molecule and a substance having a biocidal impact, then it will be required that you provide to the European authorities, in fact, 2 things. First of all, you need an approval of your biocide as an active ingredient. This is a process which is heavy. Just to have an idea, for octanoic and decanoic, just for the validation at European level, it took us more or less 6 to 7 years, and the investment is, globally speaking, 2-3 million euros, because you have to provide a full package, a full dossier, in which you have a complete analytical dossier, because you have to demonstrate that your biocide, your active ingredient, which we'll put in formulation later on, that it can be analyzed in a quantitative way. So you have to develop all those methods. So secondly, in terms of microbiology, you have to provide what they call the European EN standard evaluations. And this depends on the kind of market. So the market is split into what they call product types. To have an idea, if you go in the food market, they call it product type 4 market. And for this market, if you put a claim against bacteria, yeast, fungi, viruses, etc., you have for each specific kind of, I would say, strain, a specific EN norm. This EN norm can be a surface test, or it can be a suspension test, or it can be, in a tier 2 approach, a live test that they are requiring from you. So there is a basic portfolio of EN testing that you have to provide to put only the claim already and to say you can consider this product, this substance, as an officially recognized active ingredient. Then you have to provide a human tox and an environmental tox dossier, which is more and more difficult in the area of biocides because for the biocides, they recently decided, it's 1 year ago, you have to provide a dossier that your molecule doesn't respond to the endocrine disruptor class of molecules, of substances. So this is substantial because you need a lot of tox information, a lot of supporting information to support the claim that it is not an endocrine disruptor. And so you have, that I forgot things, but you have to provide a huge amount of information in the first step. But once that you have your active ingredient approval, you have to submit a formulation dossier, which is again a submission to Europe, and which again costs you a lot of money because they are looking to not only your active ingredient, but also to the complete formulation. And again, you need to provide those same testings, micro testing, human tox evaluation, environmental tox evaluation, etc., etc. This process is again for 4 or 5 years. So we finalized very recently our first dossier. The second will be finalized next year with a commission approval, I think in April. And we have our third dossier running with another active ingredient. So it's a huge job, but you have to know upfront the starting up of all research programs. You have to know those constraints. I saw in one of the questions, the question about the reach. Yeah, of course, in Europe, if you put a surfactant on the market, you need to fulfill the reach requirements. You can use what they call the PPROD approach, because as long as your volume is not high enough, your tonnage band is not so high, you can use, let's say, the procedure of reach for development of new substances. But there will be one day that you have to fulfill your reach requirements. So you can buy some time for 3-4 years in which you can test the molecules on the market. You have to provide a dossier to ECHA for this, but the reach requirements need to be fulfilled at a given moment, and this is also quite intensive. So it's a long process. It's a long process, and you need to, for each kind of regulation, for cosmetics, for the plant protection regulation, for the biocide regulation, the detergent regulation, you have to submit information. Yeah.
Okay, thanks for this explanation on the cost and money procedures of the regulations. Do you think about sharing the cost, the regulation cost with your suppliers, or why your suppliers didn't, we say, make it?
Yeah, there are different approaches possible. You make your economical overview, of course. Before that you do this investment, you think twice about this, because you can't say like this in a company, I need 2 million euros and we will see what the market will provide. No. So it's you have different approaches. I have seen situations in which we shared the costs even with competitors.
Yeah.
Because the cost to have the final evaluation is too high. So, and there are agreements about this in Europe and regulations about this. You have even the obligation in Europe to sit together with your competitor if you would like to buy an access to your dossier. So it's even an obligation. You have to share. There is a mandatory data sharing, and also the conditions of payment are defined by Europe. So yeah, you can share the costs with other people which are in the same kind of market with your competitors. With suppliers, this is another issue because normally a supplier and a producer of a substance, normally the market for him is bigger because if he's producing, he's selling to much more customers, like we, formulators. So the market is bigger, but at the same time for him, having a market which is bigger, the sales price is lower because we can sell product formulation at a higher price, of course, to an end user. I have seen here prices which were mentioned between 3 and 4 euro per kilogram. I would say this is already huge. For a biocide, 3 to 4 euros, so 4,000 euros per ton. This is already huge. For some specific molecules, the price could be much higher. So if you have specific niche applications, it could even be,
What type of application it could be, for example?
I give an example in water treatment. In water treatment, there are specific molecules like isothiazoline, like DBNPA, like brominated molecule substances. And those molecules are sorted at high prices on the market per kilogram. But because the end use of those molecules is in terms of ppms, you're using 50 to 100 ppm to obtain the biocidal activity. So the end concentration use is much lower. So the price impact in reality is from the supplier point of view has to be evaluated regarding your end use concentration. That's what I meant with this. So there are several possibilities. Yes, there are deals which are made with suppliers. We can make deals with competitors. I think that more and more you need to go in the directions of cost sharing, because otherwise the market will be cleaned up and a lot of small companies will disappear. And that's my opinion. And we see it already in the market. Sustainability is a cost and it needs to be shared. That's why I mentioned perhaps authorities can also put in place a stimuli for the market, like tax incentives and stuff like this.
Okay. Last questions before going to ARD. You have tested different biobased surfactants, I think. Would you accept the GMO? I would say biobased surfactants. That means surfactants coming from transgenic crops, for example. Will you accept microbiological like sophorolipids or others products?
Again, it depends of the end user. We have some big groups, and before that you are listed, they call it a positive list. Before that you are listed as an accepted supplier, you have a kind of a process evaluation which is made. And one of the criteria of, I will not give the name, of course, of this big group, one of the conditions of one of our customers is that everything should be GMO free, and you have to sign up documents for this. And so some of our customers doesn't require it, some they require it. So it depends really on how is the end user positioning, especially in the food market, his end product. If a group like Danone, for instance, say, but I'm selling yogurt and I'm claiming that this is GMO free, they are checking upfront with all the suppliers, and you have to sign up documents and you have audits. They are looking into depth in your process and your supplier agreements that everything that you're buying is GMO free. So it's up to this level of approvals. So it's variable. Some accept and some doesn't accept. Yeah.
Okay, the last question. You have tested the different samples coming from the ValBran projects and especially the antimicrobial properties. What are the first results? Is this something good or?
I have to share this with Caroline because I signed off an NDA. So I consider that I can't share this here.
Yeah, but is it good or not? Is it something?
I will not say yes or no. I will share this by respecting my legal agreement.
Okay, thanks a lot. So we are going to switch now to a well-known biorefineries research company, which is ARD and especially Boris Estrin, which will present the interest and advantage of greener alkyl polyglycoside. And Boris, the floor is yours.
To the agenda slide, and I would like to thank you for hiring your phone for me because I had some connection problem from the side of IRD, and it is a specific problem with live storm, I think. So I hope that everybody can hear me. I'm going to make a short introduction about Witoleo, in fact, because our surfactant activity is located in a daughter firm, and so I'm going to make you a tour of our activities and about industrial location of our activities in the biorefinery of Pomacle-Barencourt. Then I will talk about mainly alkyl polyglycosides. That was a type of surfactant that was of interest in the ValBran project, and more precisely about the lignocellulosic biomass-based glycoside and xylosides we are producing in Witoleo. And I will probably skip the main process option because they have been deeply introduced during the precedent presentation, not mostly by Caroline during this conference. But I will more focus on the properties, application, and toxicological differences with a benchmark chosen that is alkyl polyglycoside, that is the main type of surfactant, industrial surfactant that are located on the business area where we are the most active, and more precisely detergent, cosmetic, and crop protection products. I will end this presentation by a couple of information about environmental point of view of these ingredients, and with a couple of information extracted from life cycle inventory. So next slide, Jackie, please. Thank you. So concerning
sounds good.
Precedent slide, please, Jackie. Yeah, thank you. Concerning Witoleo, so as I told just before, we are Witoleo is a 100% owned company owned by ARD that is located in a consortium that is owned also by a consortium of agribusiness companies such as Silesia and Cristal-Lignon. And we have four main nodes that are green chemistry, wild biotechnologies, plant extraction and purification from various biomass, and also crop protection ingredients. The company is a 30-year-old company, and Witoleo has been created in 2010 in order to valorize all the research and development work we did since the beginning of 2000. And in 2010, we decided to locate all the activity concerning surfactants in the new created company Witoleo. On the next slide, thank you, Jackie. You see that Witoleo is also located on the same site of IRD on the biorefinery platform of Pomac-le-Bazoncourt, where we can profit about this privileged industrial position to have an access to various raw material coming from the main crops around us, such as sugar beet or wheat. And also we have also privileged access to energy that is produced by Crystal Union for all the platforms. Next slide. So Witoleo is also producing and selling itself this kind of surfactants, and we decided to start our business activity with alkyl polyglycosides made from xylose. Next slide. At the origin, we started producing alkyl polyglycoside from xylose from wheat lignocellulosic residues such as wheat straw or wheat bran. And then we started to widen our portfolio of surfactants by also producing esters such as glycine betaine esters, glycine betaine coming from molasses of sugar beet, but also we try to valorize also some other rates such as diesel oil, which are yielding some very interesting C5 alcohol such as ionic alcohol that can be used as building block for alkylglycosides. Concerning the production of alkyl polyglycosides in Witoleo, today we are not using enzymatic process industrially, but we are producing them by glycosylation technology. And concerning research and development, if you want to go deeper in this topic concerning chemical and acid catalyzed glycosylation, you can refer to this book where we have made a short review about all the various options you have by glycosylation to produce APG or APP by this technology, and for example making alkyl polyglycoside without any acid catalyst, for example, is something that is possible. And we have also make a lot of research and development work on this area. On the next slide, you see the conventional route that is used today to produce alkyl polyglycoside, more or less the glucose that is used in the industry by the other companies than Witoleo who are producing alkyl polyglycoside. They are using glucose that is mainly coming from the food industry that is producing glucose for drinks or for sweeteners, etc. And the new approach we have developed within Witoleo was to use a cocktail of sugar coming from lignocellulosic biomass. So we started to produce them from wheat, and then we have decided to also collect xylose from other botanical sources. On the next slide, concerning enzymatic technology, we had before ValBran project had a very good cooperation already with the teams of Caroline and Richard concerning the production of alkylglycosides. And at that time, after being involved in the filing of some patents with our partners from the university and from INRA, we have not decided to go deeper in the design of an industrial process by this technology. The main drawback at that time was the yield of surfactants and the productivity of this enzymatic process that was not enough and not high enough to imagine and envisage industrial production of these surfactants for an industrial production and a good opportunity to sell them to the detergent market or cosmetic market. Next slide. So what is the main interest, in fact, for having lignocellulosic sugar-based glycosides in the portfolio of wheat oil? I would say that on the next slide, you have the main advantage of having xylose in the polar head of your surfactant. By the way, you have the opportunity to have surfactants that more or less are more performant in most of the application and in most of the properties of a surfactant. By giving a lower solubility to the surfactant, the HLB is lower, the critical aggregation concentration are lower, so that end in some higher efficiency in detergent activities, for example. And also you have better surface tension lowering, giving you a more performant surfactant during detergent activities. All these intrinsic properties of the surfactant and also in some better efficiencies in some performance tests that are usually done by our customers in detergents or in cosmetics, that are the degreasing index, the wetting power, etc. And in all these type of application tests that are normative tests done by our customers, alkyl polypentoxides were performing better than alkyl polyglycosides. On the next slide, I would say that the second advantage of having this type of surfactant in our portfolio is that alkyl polypentoxides, also alkyl polyglycosides, are very soft surfactants. The pentoxides are softer. This has been displayed by all the in vitro testing we have done in view of the filling of the REACH dossier. We have first collected some bibliographic data in the open literature concerning cytotoxicity test, where xylose-based glycosides were very more softer than glycosides in terms of skin irritancy and eye irritancy. We have also performed internally red blood cell test. That is the second in vitro test that is the most used by the industry to assess the potential irritation power of this surfactant. And we have also proven that our alkyl polypentoxides were much more compatible with skin and eye than APGs that are also not very irritant surfactants. On the next slide, you have also the confirmation of the in vitro testing by the in vivo testing that was at minimum required to fill the REACH dossier. And all these tests in our end, the output of that is that on the safety data sheet of the products we are selling that are composed of xylosides have a lower labeling concerning the risk phrases. And you have on this slide the differences between an APG that is potentially irritant when it is considered at 100% of active matter. The pentose containing glycosides are lower in terms of irritancy, and that allows all the customers in the application where the global harmonized labeling is considered to have more concentrated products in terms of surfactant concentration and without any risk phrases or hazard label on their product. So that's the main two advantages advantages of having APP. And if you then go to the environmental point of view of this surfactant, on the next slide you have an indication of what the life cycle inventory is giving concerning ATP of the same medium chain alkyl tail length than an APG. You have a lower consumption of fossil energy, for example. And here on this slide, it is displayed striking differences with some alcohol ethoxylated, sulfated, for example. And on the next slide, you have a figure that is very important that has been produced by a complete ACV that we have done and published in Green Chemistry Journal, considering the production of alkyl polypentoxide from wheat straw in a biorefinery environment. And we have compared this scenario of production with a reference scenario that is the production of alkyl polyglycosides from glucose coming from the food industry. And in all the categories of this ACV, you have a saving of the impact of your surfactant production for global warming, from the waste you are producing, etc. In all categories, alkyl polypentoxide displayed a lower environmental impact in all the aspects of the process. So that is the third and last very important advantage of having pentose in the polar head of surfactants, and in the way you are producing them in a biorefinery context, where you have a privileged access to the lignocellulosic biomass, and you can use it on site and valorize all the products internally of the production of the surfactant. So that's the last slide of my presentation. So I was set on a 15-minute presentation as required by Laurent at the beginning of the contact we had for the ValBran project. But if you want to have more information, if you have questions, I can answer today. But if you want to go deeper in the topic of one of the topics of this presentation, don't hesitate to contact me on the mail address you have on the first slide of the presentation. And I'm available for all the questions you have today. Thank you very much, Jackie, for the logistics and for passing the slide for me. Thank you.
Okay, thanks, Boris, for this nice presentation on really the advantages of the alkyl polypentoxide, not only the technical and environmental, I will say, advantages. A question: you have taken something like 10 years for the development of the wheat oleo range of products. What can you say about the industrialization or commercialization barriers today for bio-based products?
Yeah, it didn't take 10 years, but we started with oleo in 2010, when we were ready to produce industrially the first line and types of surfactants we were aiming at putting on the market at that time. So we were already ready at the level of industrial volume potential. And the main barrier of developing this business, first of all, is to have some important customers that are not only customers but also partners during the first years of your business implementation, because they are going to validate all the industrial specification of your ingredients, and then you need to also be sure you are going to accompany the industrial development of your surfactants by a lot of capital expense, because today we have a production facility of around between 2 and 5 tons per year of surfactants for all the portfolio of surfactants we propose with Oleo. And we started with an on-stream capacity of around 500 tons per year. So that means since 10 years, we are a small company, so we have some limited resources, but the CapEx is very important to develop this business. And then obviously the relationship with your customers is also very important. So you have to connect with them at the good connection point, because you need to have a connection with the research and development in the first steps of the sales, and then you also need to have a good connection with the buyer of this company when they are well organized with the buyer, and for big group it is the case obviously, and you need to have a good connection with them. Okay,
you mentioned that the capex is very important. That means you invest in a new.
And obviously, when you put on the market, you are obliged to have the rich dossier, and all the required regulatory aspects. So when I say that you need to validate with your customer all the figures in your safety data sheet, it is the point. You need to have done all the tests required for the application of your customer.
Okay. You mentioned that you didn't switch to enzyme process first because of the yield. Can you give us some comparisons? What is today's glycosylation yield today, for example, and how you compare it to biocatalyst way?
I would maybe make a short comment about the precedent presentation concerning the cost or price that have been indicated between 3 and 4. Today, the business value of alkyl polyglycosides, APG and APP, is ranging between 1 and 2 euro per kilo. So that is the price. And then, as we are not a company that want to lose some money, we need to earn money. So we need to have cost of production that are lower, obviously. So this is the main figure to do at the end of people who are in the audience of this conference today, is that the price needs to be in this target to be sure that we are well business oriented and profitable at the end. Concerning the enzymatic process compared to a chemical process, the chemical process is yielding, you have a conversion of the sugar during the glycosylation or transglycosylation process when it is conducted with chemical catalyst. You have a yield of at minimum 95%, and it range between 95 and 99, quasi quantitative in fact. And the second point is that it is also something that was not commented during the presentation, is the productivity, because the higher is the productivity, the lower is the capex you need for reaching on very high on-stream capacities. And these very high volume production capacities, you need to add them to knock on the door of the big players, on the big customers that are going to require to their supplier a very good volume capacity to make them sure that they can buy all along the coming years the good product to you. So it is very important to have a very good productivity. And concerning the yield, yes, the yield is from the sugar introduced in the process. So I thought that from the ValBran process, you have a yield that is quite good, but it is something that is raised to 80% today. But when we consider the biomass residue that is going to be introduced, you need to have a complete process and design of the process that allows the valorization of all the parts of the lignocellulosic residue. So what are they going to do with the lignin? If there is some lignin in the biomass used, what is about the protein maybe, if it is present. So you need to have a complete overview of what is going to be done with that, or with the other part, the non-sugar part of the lignocellulosic biomass, and you need also to assess what is the price of the biomass at the entry of the plant, because today it is not a zero value biomass. It is something that is sold to other applications, so you are in the competition with your way of valorization. So the surfactants industry for detergent, etc., you are in competition with other markets such as energy production, with some feed application. So you need to have also a good idea of what is going to be the price of the lignocellulosic biomass at the entry of the process. So it is very important to have a very good and secured sourcing about that.
Okay, last question before going to the conclusions of the webinar. Do you see a modification of the market post-COVID, and which new market could be interested in biobased surfactants? We speak a lot about cosmetic and detergency, not a lot on plant crop protections. So how you see the biobased markets in the futures?
So what I can say, I'm not very good to see what the future is going to be. But, what we were sure when we started Witolio was that the future is with sustainable surfactants in all the applications such as detergent, cosmetic, and also maybe crop protection if we continue to have a good agricultural politics in Europe, because we need lower irritant products made from vegetables, renewable raw material. And when we started this industrial activity, there was main step in the first year of the development of this activity. The first one was the introduction of the new CLP regulation. The president, so Pura mentioned the real importance of the regulation in their applications. For us, it was also a very key, important steps when the CLP was introduced in 2016. It obviously it was a good, important way for customers to select preferably some surfactants in the area of renewable raw material, but not only because they are renewable, also because they are less toxic, etc. So it was the health topic that was on the first with the CLP regulation introduction. Concerning the COVID, today I would say that COVID is just strengthening what we are seeing for 2 to 5 years. Just giving not new way of developing products. It is just giving rise of and new opportunities for this kind of products in all the industrial application I was talking about. So I would say that it's not a key step for us and not a big bottleneck today. Just lowering of the consumption probably is going to be ended in a couple of months or year, but probably we are going to start again the growth for Witoleo after this period. So I would say that it's not going to do something different than before COVID. Customers are continuing to look at surfactants or other ingredients that are renewable, less toxic, more performance, more efficient, and at the overall, I would say at the same cost or equivalent cost of the cost we have today, depending the evolution of the economics, obviously, and financial topics that are external things that we can't be active on. But today, I would say it's not going to change something at the time.
Okay, thanks, Boris, for your explanation. So now I will give for the conclusions the floor to Caroline Raymond.
Thank you, Jackie. So yes, now it is time to conclude. So before the conclusion, for us, it is important to evaluate the impact of our final event. And so we ask you to give your opinion about this event with an online survey that will take 2 minutes to fill in. So please, before leaving the webinar, don't forget to fill in the online survey by clicking onto the icons, which is called sondage, as shown on the slide. So to conclude, this slide presents the highlights of the project, of the ValBran project. So the project involved 9 partners, which strongly collaborated. During the project, 4 postdoctorates, 1 engineer, and 3 technicians were recruited. In terms of valorization and communication, after 4 years of project duration, 3 scientific articles were published and 2 others are under submission. 27 communication actions were realized. 185 companies were informed about the project ValBran during the different events that were organized, and one final didactic report was prepared. I will come back onto this report in a few minutes. And finally, some biobased surfactants and also the ValBran residues obtained at the end of the enzymatic process were delivered to 4 companies for testing. So about the final didactic report, it will be available under paper and online version. The online version will be soon available from mid-December, and you will be able to upload this online version of the final didactic report with the internet website www.witbransurfactants.eu. And it will be available in French and in Dutch. So don't hesitate to go onto this website from mid-December. And so finally, I would like to thank all the speakers and all the participants of this final event. I would like to thank Jackie from IAR Cluster for managing the moderation of this event. And I would like to thank Laurent Debatty from Valbiom, who organized this webinar. So thank you for your attention and goodbye.
Goodbye.